right, we are going to talk today about mutations. As a general recap, we last semester talked about protein synthesis. So, how we take the DNA, make RNA, and make the subsequent proteins. And then last time we discussed mitosis and meiosis. So, what happens when cells divide, whether that be somatic cells using mitosis or gametic cells using meiosis. And so now we're sort of going to backtrack a little bit and talk about mutations. Now, the mutations we're going to talk about today, we're going to talk about two different categories. We're going to talk about mutations that happen at the DNA level, meaning way back when we were talking about protein synthesis, what happens when that DNA isn't correct, or it isn't the DNA that we intended it to be, and how that would ultimately impact protein synthesis. And then we're going to talk about sort of more detrimental mutations that affect like a whole chromosome. And the reason why chromosomes obviously hold more genes. So when you have a DNA mutation, that mutation is only going to affect one gene, whatever one you're coding for. But when you're looking at the chromosome, because there are multiple genes in that chromosome, depending on what type of mutation it is, you could have all sorts of different things that could happen because there are multiple genes involved in a chromosome. So we're going to talk about both today and sort of loop it under this umbrella of mutations. So I'm sure you know what a mutation is. A mutation is just a change in the sequence of a nucleotide, right? Like if you had the sequence TTTTT in the DNA and you had TTTTA, that's a mutation. Now, whether or not that DNA mutation is going to affect the ultimate protein or not, that's where all of that, our knowledge about protein synthesis comes into place. So on the smaller end, we're talking, one single nucleotide change. On the major end, we're talking about a whole chromosome. So we're going to start with DNA mutations. So this is mutations that are happening at the DNA level. So we'll start with the idea of a base substitution. And that's exactly what it sounds like. In base substitutions, you're substituting one base for a different base. So where there's supposed to be a C, G got substituted instead. So as you can imagine, if you have the sequence different, when you go ahead and you make that into a protein, the sequence could result in a different amino acid. And we'll talk about that in a second. When you have a base substitution, there are several possible outcomes. The first is one that is probably more common than we would ever know, and it's called a silent mutation. And it's silent because you don't actually see any sort of impact on the resulting protein because you are making the same amino acid. So if you remember from protein synthesis, we talked about how there are 64 codons, but there are only 20 amino acids. And when you looked at that big wheel, there were, in some cases, 4 different codes for the same amino acid. What that means is that if a base is substituted, it could be substituted with another nucleotide that would code for the same amino acid, in which case there would be no noticeable effect on the protein. There would be a letter different, so it's a substitution, but it's silent because you don't actually see anything happening in the protein. A nonsense mutation is when that base substitution creates a stop codon somewhere in the middle of the sequence. So instead of the stop being at the end of the protein, what we have in a base substitution is that letter gets substituted into a code that gets read as stop. So the protein ends up being stopped prematurely. A missense mutation is a base substitution that happens that replaces a letter, but in this case, unlike a silent mutation, it makes a different amino acid. And because of that different amino acid, you end up with a different polypeptide, a different protein. We see this in sickle cell anemia, where one base change in hemoglobin, which is the protein that carries your oxygen, actually results in making it's a missense mutation. It's just one change results in making hemoglobin that is no longer effective. And so we do see these different diseases that are actually base substitutions that end up being missense mutations. So here's this chart. I'm going to grab my pointer. Here's this chart. So if this was the original DNA that we wanted to, that we were expecting, so TTC was the codon, a silent mutation would end up creating the same amino acid. So you can see here, when we go to make the protein, we end up with lysine. So it's you wouldn't know that this was not supposed to be here. In a nonsense mutation, what happened here was this first T was substituted with an A, which ended up coding for a stop. So we prematurely stopped it. Don't worry too much about conservative and non-conservative. Just notice when we change these two, we ended up with a different amino acid than what was intended. That's a missense mutation. The second type of mutation that can happen is you can actually have a base that's inserted, so an extra base, or a base that's deleted. So imagine if you had a sentence and someone threw in an extra letter and you had to read that sentence in threes. Everything's going to shift over. So now you end up affecting all the amino acids after where that letter is inserted or deleted, because everything's going to shift over. So the shifting over, that is called a frame shift mutation, because the entire reading frame, because remember, we have to read it in threes. So if you add a base or delete a base, you're going to shift that frame over and all the amino acids are going to be affected after that. Usually we end up with a non-functional protein because you've literally changed all the amino acids and not just one. So when we talk about mutation, what happens? Why does this happen? Why do we end up with the wrong base? And so, let's stop here for a second and just show you this. So here was what I was talking about. So if this is the normal DNA, you can see here between the A and the C and an insertion, we inserted this extra C. That's going to change the reading frame. So instead of the amino acid being CAA, it's going to be CCA that we're going to code for eventually. ATA, it was TAC. So you altered it this way. When we deleted it, we shifted it this way. So now we've shifted this from GGA to GGC. So when we talk about mutagens, we talk about things that are actually going to change the DNA. We talk about things like radiation. So when you hear of people talking about UV radiation being a carcinogen, that's sort of what we're talking about there, is this idea that it can alter your DNA. So when you think about DNA mutations, it's oftentimes something is mutating that DNA. All right, so now we're going to jump over to chromosome error. So what happens when it's not just happening at the DNA level, but it's actually happening to an entire chromosome? So that's what we're going to jump into now. So if you remember in mitosis and meiosis, the goal in mitosis was for those chromosomes to separate and to produce two identical cells with the same number of chromosomes. And the chromosomes were identical. In meiosis, we had this situation where the chromosomes were supposed to separate and then separate again. So we end up with 4 non-identical cells. So the genetic material is different between the 4 cells. That's ideal. That's what's supposed to happen, is that at the end, you get 23 chromosomes from the maternal side, and you're going to have it fertilized by 23 chromosomes from the paternal side. When we look at chromosomal errors, we see these in meiosis. So what happens when those chromosomes don't necessarily split properly, or what happens if we're missing part of it, things like that. So that's what we're going to talk about right now. So the first type of thing that can happen is something called non-disjunction. And what happens in non-disjunction is, instead of those chromosomes separating out, so you get 23 and 23, you end up having a chromosome that sticks to its homologous chromosome and traveling into the cell together. So instead of splitting, they end up together. And what ends up happening there is you have a cell that ends up with 22 chromosomes and one that ends up with 24 chromosomes. So if the cell that with 24 chromosomes get fertilized, remember there's going to be, so let's just say that the 24 chromosomes comes from mom. If it gets fertilized by a normal sperm with 23 chromosomes, that fertilized egg will now have 47 chromosomes instead of 46 chromosomes. There are some chromosomes that can be experience trisomy, and the baby can continue and live, and there'll be some issues and developmental issues with the child along the way. The X and Y chromosomes can have trisomy happen, and there are impacts on reproduction and sort of those parts of your existence, but otherwise you lead a fairly typical existence. But most trisomies are actually not conducive to life, meaning that the fertilized egg would abort, because it's the trisomy doesn't offspring with trisomy don't necessarily make it, but there are a couple of chromosomes that's possible. But like trisomy of chromosome number 1, they wouldn't survive, for instance. So it doesn't happen very often, in the sense that those babies don't necessarily make it past 6 or 7 weeks in development. So these can be miscarriages that happen very early on in pregnancy. And it's usually because there was some sort of issue with the chromosome number. So this is sort of what a trisomy could look like. If it happens here in meiosis 1, this would be the situation. So this would actually create eggs or sperm that would not result in a fertilized egg. It would not work. Here, this is more likely to be a situation where you could potentially have a baby that could survive. And here you have this fertilized egg here that has 2 chromosomes. This one has 0. And again, this would be a specific chromosome number, right? Like number 4. So 2 chromosome number 4s, and this one has 0. So the trisomy comes because it's 2n plus 1. So it ends up being 3n. And again, the non-sex chromosome disjunctions are usually not conducive to life. Yes, there are people who live with trisomy 21, trisomy 18, and trisomy 13, but a lot of them don't make it to birth. So we talked about a karyotype before, and we looked at this before. And so when you go in for prenatal testing, they can run a karyotype, and they could see, for instance, oh, you have 3 chromosome 13s, which would be trisomy 13. You'd actually be able to tell that from a karyotype. You can have trisomy in the sex chromosomes, and those usually are conducive with life. Turner syndrome is an example of trisomy, except for this is actually monosomy. So this is an example where, and I'll go back here so we can see it while we talk through it. This is an example where this egg is fertilized and it's missing an X chromosome. So it's missing the X chromosome from either the female or the male. But the male, so let's say it's the female's missing the X chromosome, but the male has the X chromosome. So technically speaking, that female is XO. So they are female because of the X, but they don't have both X's. And that's Turner syndrome. If a sperm it has the Y chromosome, and the the egg is missing the X chromosome, and they fertilize each other, and you end up with a YO, that actually will not make it. You have to have an X chromosome in order to survive. So here is somebody with Turner syndrome. As you can see, there's no X chromosome here, and there's no Y chromosome that would make it biologically male. So this person has monosomy X, which is Turner syndrome. So that was non-disjunction, when a whole chromosome doesn't separate out. But there are conditions in which you are missing or you have extra pieces of chromosome. So it's not the whole chromosome, it's pieces of the chromosome that are have some issues. So in deletion, you lose a piece of the chromosome. The most famous of that is there's a chromosome number 5. There's a deletion that can occur in one of the arms of the chromosome, and it results in something called Cri du chat, which means cry of the cat. And so there are conditions that are associated with these specific deletions. In duplication, what happens is one chromosome piece kind of just gets stuck to another sister chromatid. So it's kind of like a non-disjunction, but it's only a piece. And so it doesn't necessarily result in anything that is too detrimental, because all the genetic information is still there, and it's not, it doesn't affect the offspring. Inversion is when a piece of a chromosome breaks off, flips around, and reattaches in the opposite direction. And that looks like this here. So inversion here. So I love this graphic because it's really helpful in seeing all of these. So deletion, you can see this one part of the chromosome here is just deleted. So again, this is not as detrimental as maybe a non-disjunction would be. In duplication, you can see here this section, the section of DNA here just got repeated over here. So it's not, again, it oftentimes duplications don't result in anything that's really noticeable in the offspring, because the information is still there. An inversion here, this should be helpful, hopefully. You can see here BCD. So this broke off, flipped around, and now it's going in the opposite direction. So DCB. So it's reading backwards. We'll talk about this I'll talk about it right now because the picture's up. So a translocation. So these are non-homologous chromosomes. So these are not, chromosome number 1 and chromosome number 1. This could be like chromosome number 1 and chromosome number 7, let's say. So what happens is a piece of chromosome number 1 and a piece of chromosome number 7 break, and they swap. So you ended up affecting 2 separate chromosomes with this switch, because you ended up switching these portions of these chromosomes. And because they're not homologous, because remember, in crossing over, we want that to happen, right? We want homologous chromosomes to swap information. We don't want non-homologous chromosomes to swap information, because it's not the same information. So you end up affecting 2 chromosomes. So that's translocation. And again, here, this is showing you all of this. Reciprocal translocation just means it's the same amount of information between non-homologous chromosomes. So most chromosomal changes that happen when we think about cancer are somatic cells. So if you have a cancer in the somatic cells, you're not going to pass it off to your offspring, because it's in your somatic cells, not in your gametes, your egg and sperm. All right, so that is mutations, kind of a quick view of it, and there'll be some follow-up activities to hopefully cement this. But this was kind of a shorter lecture because the information is pretty cut and dry. And hopefully this helped, and if you have questions, let me know.